everyone, this is Sharmya. In this video, we are going to see about the replication of virus. First of all, introduction. The virus are the obligate intracellular pathogens. They cannot replicate without the machinery and metabolism of a host cell. They require a host cell for replication. Host cells mean living host cells like humans, bacteria, or fungi. They need a living host cells to replicate. All viruses are depend upon the reproduction and metabolic processes. By themselves, virus do not encode any type of enzymes necessary for viral replication. They use their host cells to replicate. They use their host cells enzymes for replication. But within a host cell, virus will act as a commenter. During the cellular replication only, the virus also replicate, so that they will act as a commenter. Phages replicate only in the cytoplasm because they do not have any nucleus and all, so they will replicate in the cytoplasm area of the cell. In eukaryotic cells, most of the DNA viruses will be replicate inside the nucleus. That means inside the DNA only they will start a replication. RNA viruses that infect the animal cells mostly replicate in the area of the cytoplasm. And although the replication life cycle of the virus differ from the species and category of the virus, there will be 6 basic stages of lytic cycle. Overall, the viruses can replicate like a lytic cycle or a lysogenic cycle. In actual, viral replication differs from species and the category of the virus. I have already told that there are 6 stages of viral replication. First one is attachment, second one penetration, third is uncoating, replication, assembly, and virion release. Virion is a mature form of the virus. First step for viral replication is attachment. For attachment, the naked virus will attach to the specific receptor which was found in the host cell, so that there must be a specific receptor which the virus is attracted. According to Lee, many viruses can only infect a limited range of cells. For example, HIV virus will infect only the T helper cells that express the CD4 and the CXCR4 receptor molecule. So that some viruses only can infect the our host T helper cells. So they are most of the viruses are host receptor specific viruses. After attachment, next step is penetration. After attaching to the specific receptors, the next step, the virus will penetrate into the host cell. It will inject its nucleic acid, DNA or RNA into the host cytoplasm. Then after reaching the cytoplasm, it will enter into the host nucleic acid. There are 2 different forms of penetration or entry of the virus. For enveloped virus means for cytoplasmic membrane fusion, the virus will fuse into the host cell cytoplasmic membrane, and after the fusion, the viral contents will be released into the cytoplasm of the cell. The second one, endocytosis. During this, the host cell cytoplasmic membrane will engulf the virus and take the virus into it. For naked virus, the virus will passes into the host cytoplasmic membrane. The third step is uncoating. After penetration, the viral will remove its capsid and degraded by the degraded by the viral enzyme, and it will release the nucleic acid, means DNA or RNA, into the host cell. During this stage, it will start infective. During attachment and penetration, we will not show any infected symptoms, but during this uncoating stages, the people will start gaining of signs and symptoms of viral infection. Next step after uncoating is viral replication. After uncoating, the DNA of the virus and nucleic acid and proteins of the virus has to be replicated. There are two stages for this kinds of replication. Virus will start a replication means it will cut off its DNA into two particles, and half of its DNA will attach to the half of the DNA of the our host cell. So it will join and make the new DNA. Then other half and other half of the host will be joined to make the head, tail, and cell membrane proteins of the viruses. After that, the virus will completely replicate. Next step is assembly. Viral proteins will packed into a newly replicated they will pack into a newly replicated genome into they will then converted into a new virion. Virion means matured virus. This process is called as maturation. The last final step is release of the virion. First of all, there will be two methods of release. One is lysis or budding. Some viruses after replication, they will kill the host cell and directly released into the blood. Example, smallpox virus. This is referred to as cytolytic viruses. Some enveloped viruses like influenza A virus, they won't kill the cells. They will release by the step of budding. These are not usually kill the infected cells, so they are termed as cytopathic viruses. After virion released into the blood, some viral proteins will remain in the host cell membrane. They will act as a potential target for the circulating antibiotics. The circulating antibiotics will target this infected cells only, which has small amounts of proteins. Other viruses will be released directly into the blood. So we require antiviral medications if we are infected by a viral infection. That's all. Thank you.